Okay, well, let's get started again. We're going to bring Sarah back to the stage, and she's going to talk about what is happening with BBSOAS research. Thank you.
All right. Let's see. I don't know how to switch this to show the slides. Oh, great. Perfect. Okay, so for this second talk, I am going to be talking about a project that I've been working on over the past couple months. It is still an ongoing project, but the goal for this is to identify everything that we know about NR2F1 already, who are the people who are studying the gene, what resources they have, what we as the patient advocacy community have, and then what are the gaps, what are the big questions that we still have about how NR2F1 works in both the healthy condition and how it results in BBSOAS, and so what is it that we need in order to be finding treatments. So I'm going to be giving just some excerpts of a much larger presentation that I'm working on, and I might be skipping some slides for the sake of time. I think I have way too many for the amount of time that we have, but I'll be happy to answer questions some other time about it if you want to know more. So we're going to dive in. So this was just the timeline that I showed in the past talk, kind of identifying all the things that we need in order to get treatments approved, ranging from first identifying the gene, then also natural history data, biomarkers, outcome measures, models of the disease, etc. So here is kind of the overview of what I'm going to be talking about today. And again, some of this I might kind of skip through just for the sake of time. And this is still also a work in progress. So starting with just kind of the overview of what BBSOAS is. As you all know, it's due to a mutation in or a deletion of the NR2F1 gene. So typically these are de novo mutations, but sometimes they are inherited. These can be nonsense or missense or frameshift mutations, as well as deletions. And there have been 92 published patients in the literature, but close to 200 identified people in the world. And based on some estimates, we believe that there may be as many as like 3,000 to 4,000 people in the world with BBSOAS who just have not been diagnosed and identified yet. The clinical presentation of BBSOAS, as you are aware, can range pretty across a pretty broad range of symptoms, from relatively mild ones, typically with from deletions of the gene, to much more severe phenotypes due to mutations in the DNA binding domain and everything in between. But typically there's developmental delay, both in terms of motor skills and speech and other cognitive abilities, hypotonia or low muscle tone and resulting coordination problems from that. Visual impairments of all sorts and abnormal eye movements are also common. Hearing impairments have also been reported. Seizures and epilepsy, autism and other behavioral problems, and love of music and exceptionally great memories are also reported for BBSOAS. Right now there is no treatment yet to treat the underlying cause. It's so far just symptom management. So we want to work on finding something more targeted for BBSOAS. Then again, here's some data on what we know about some of the most common phenotypes. This comes from a literature review, but the data will match up pretty strongly with what I believe Katya will be showing later with the data from Matrix. And so that's always great to see that there is an alignment between what has been reported by clinicians and scientists in the literature and then what you as the families are reporting. Then for the genetics of it, so mutations are seen across the entirety of the NR2F1 gene. So this here is a schematic from one of Dr. Schaaf's papers showing where these mutations have been identified. So there are many at the translation initiation site, so that affects whether the protein is made or not, as well as a lot of the mutations are in the DNA binding domain area. And then more mutations have been identified in the ligand binding domain over time as well, because these patients typically have a more mild presentation that oftentimes those patients, the milder ones, are not identified until the disease has been known for a longer amount of time, just because having the milder phenotype means that there's just not as much push to get them diagnosed with a genetic cause. But over time, we'll probably be identifying more and more of these individuals. And then in addition, there are individuals who have a deletion of the entire gene. And there are actually a couple different NR2F genes. So there's NR2F1, NR2F2, and NR2F6. So these genes have a lot of homology, which means that they have similar sequences and the structure of the protein is similar, but their functions can be different. These are probably all transcription factors, but we don't know exactly what each of them is doing and if they may be interacting with each other. So that is still an area of research. Regarding the mechanisms of BBSOAS, we know that NR2F1 as a transcription factor is able to both increase expression of some genes, so turning some genes on, as well as turning other genes off. And so via this function, it's able to really just kind of regulate and fine-tune the expression of many other genes that are necessary for brain and eye development. And the NR2F1 gene and protein probably has multiple other partners that help determine whether it is acting as the on switch or the off switch. So then when there are mutations in NR2F1, multiple different things happen. So because NR2F1 is likely forming either dimers with itself, so just partnering like two of the protein together, or forming heterodimers with other proteins, then when you have a mutation in NR2F1, that can be affecting the way the protein is able to interact either with the wild type NR2F1 or with any of those other proteins. And so this would interfere with its ability to act as that on-off switch for other genes. So we know that then for the deletions, it basically just means that there's 50% of the amount of NR2F1 as there should be, and versus some of the other mutations where protein is being made, but it's this non-functional protein, then that can be just pairing with the wild type and interfering with the ability of the wild type protein to do its job. And so instead of having just 50% of a good copy, you then have another half of that good copy is being prevented from doing its job by the dominant negative effect of the mutated NR2F1. And so this results in even less available NR2F1 to regulate those other genes. And so this is resulting in dysregulation of many other genes in the brain. And going into what sort of mutations have been identified so far. So this here is, again, a snapshot from a paper from Michelle and Christian, as well as a literature review that one of the interns at Combined Brain helped me with. So this was looking at the different types of mutations that are seen in BBSOAS. We can see that around half of them are missense mutations. That means that a single amino acid has been changed, just basically like one letter in the sequence of the protein. And then there are around 20% are deletions, and then 10% are translation initiation site changes, and so those are going to be similar to the deletions, as well as then another 25% or so that are the nonsense or frame shift mutations, and those can have differing impacts on the protein. And so we know that NR2F1 belongs to this family of nuclear receptor proteins. So these are proteins that hang out in the nucleus of the cell where the DNA is, and then get activated by something else in the cell to trigger either that the turning on or off of the genes. However, we don't know what that switch is, what it is that activates NR2F1 in the cell. And so that means it's called an orphan receptor, because like the NR2F1, it's a receptor, but what is interacting with it? We don't know. The endogenous ligand, as its term, is not known, so it's an orphan receptor, as are NR2F2 and NR2F6. It is known that these NR2F proteins will form homodimers or heterodimers, so either interacting with each other or interacting with other proteins, and that these dimers are what is binding to various regions of DNA to turn gene on or off. We know that NR2F1 is expressed in both the peripheral and central nervous system during development, but less has been studied about NR2F1 in the adult brain and how it, what role it plays in the developed nervous system. And then getting into some of the existing resources that researchers can use for drug development, starting with the natural history. So on Matrix, there are 126 patients when I last checked, and 23 participants in the biorepository, which includes a couple siblings who have also participated to help out their brother or sister with BBSOAS. So I want to give a thanks to any of those siblings who might be in the room. And all of this data that is on Matrix is accessible to researchers. They can request it and be able to look at everything that you have filled out about your child. But currently there have not been any requests from researchers. So this is one thing that we as a community can be doing, is helping to just raise awareness of this data and see if anybody might be interested in using it. And then there are also natural history studies being done by clinicians, including Dr. Viral Shah here in the US, and also our own Dr. Christian Schaff in Germany is doing a natural history study of BBSOAS as well. So we have multiple natural history studies ongoing between the caregiver reported stuff on Matrix and then the in-person clinician side of things in both the US and in Europe. And looking at then some of the models that we have, there are in vivo, which means like living animal models for BBSOAS, and in vitro models. So in vitro is like cell-based. And additionally, then we have patient samples. So with the in vivo models, there are multiple types of mouse models that researchers have. So Dr. Michelle Suter and Christian Schaff both have a whole bunch of mouse models. They have a model of the NR2F1 deletions in mice, as well as models of the DNA binding domain missense mutations and a truncation in the ligand binding domain. And then there are efforts underway to have another mouse with the ligand binding domain mutation. Additionally, there is a researcher in China who has a different mouse with a DNA binding domain mutation. So we can see that multiple types of BBSOAS patients and phenotypes and mutations are being modeled across these different mouse models. Then Michelle also has some really cool mouse models that can be used as tools to study like specific types of neurons and tissues. And a researcher at Baylor also has some of these. And so this can give us insights into specifically like the visual problems or other questions like that. In vitro models, there are a bunch. So some of these are the iPSCs that Michelle and Christian and Magdalena have. So this is 6 different mutations as well as the knockout mutations. And there are other cells used by other researchers as well in different cell types can tell us different information about the role of NR2F1 across multiple tissue types and developmental time points. And yeah, more here. And then Michelle Suter also has something called an organoid, and so that is starting from the iPSCs, those stem cells, and then growing in a petri dish a like miniature tiny brain to again kind of understand what it is that NR2F1 is doing in the development of the brain. Let's see. All right, thank you. And then getting into the patient samples that we have from the community, we've collected multiple PBMCs, and so this is a vial of blood that will be used to create iPSCs, the induced pluripotent stem cells that can be used for all sorts of cool research, including the organoids and other work as well. And then regarding plasma and dried blood spots, whole blood, and urine, there were too many for me to list on this slide, thanks to all of the efforts from you guys from the past year. And we're hoping over the next 2 days to get even more, so then I definitely won't be able to list it all on one slide. And so this is just fantastic to have these samples here. All right, and then current therapeutic strategies for BBSOAS. So there is a compound that was identified from a researcher who had actually no idea about BBSOAS. He was studying NR2F1 in a completely different context and was trying to upregulate the gene and identified a compound that was able to do that. You know, it was increasing the expression of wild-type NR2F1. And so then Dr. Kyle Horning, who was a previous postdoc at Combined Brain and had worked with the NR2F1 Foundation, discovered this paper and reached out to that researcher and was like, hey, that could be really helpful for BBSOAS. And that researcher was excited to work on a project. So that was Dr. Julio Aguirre-Guizo at the Einstein College of Medicine. And so he's been now collaborating with Michelle Suter and Christian Schaff and Viral Shaw, trying to put together a grant. This is the R01 that Melissa mentioned for the National Eye Institute, trying to get funding to take this from very early preliminary research, looking at NR2F1 in completely different organs and disease states, and try to look at more BBSOAS specific uses and see if it might rescue some of the phenotypes in mice and then go from there. And then regarding genetic strategies, there are some possibilities for BBSOAS because we know that knocking down the mutant allele should result in a milder phenotype. We know that people who have just a deletion of NR2F1 tend to have milder symptoms. So for those of you with the DNA binding domain mutations, if that protein could be effectively just shut off, then it could look more like just the deletion symptoms. But then regarding trying to increase the amount of NR2F1, the wild type version, through genetic strategies, that's something that is going to take a lot more basic science before we're there yet, but work is being done there. And yeah, some of that work is being done by Christian here, looking at the ASOs from Ionis in iPSCs and I believe in some of those mouse models. All right, and then getting into some of the additional stuff that I've been doing as part of this project is trying to identify like specific points that whether it's the researchers or clinicians or the patient foundation and community can be doing to help further the research. So again, here's that timeline, and then I tried to identify various things along that timeline that could be helpful. So this ranges from things in green that are already being done, And then ones in yellow are things that are in progress. And ones in red are areas that we could start to look. And then ones in blue are the ones way down the road with the FDA and such. So I'm going to kind of get into some of these. And things are broken up into different categories here. There are the patient engagement projects, so that's ones that all of you can be directly involved in, and then collaboration projects that would be for looking at the researchers and pharmaceutical companies, funding projects that all of the fundraising could be done, to then help to give seed grants out to researchers to jumpstart new projects and general strategic decisions. So one way to have a more engaged patient population is you just need to find the rest of the patients. As I mentioned, based on some estimates, there may be a couple thousand people with NR2F1 mutations on the planet right now, but they have not been identified, in part because NR2F1 is not on all of the panels for sequencing. So some of you, when you got the genetic testing for your child, may have done a panel. And so what that means is that the sequencing company was only looking at a specific number of genes, the genes that they thought were the most likely to be responsible for whatever symptoms your child had. They did not necessarily look at every single gene in the DNA. And so we would then want to add NR2F1 to all these panels that might be relevant. So for one of the big companies, for example, Invitae, they do a bunch of these panels, but NR2F1 is only on the neurodevelopmental disorder panel. It is not on ones for epilepsy or for like brain malformations. So if any of your children have like abnormal MRIs, that is a really important piece of information, but that wouldn't be something that would, if that was sent out for sequencing, wouldn't show up on that panel. And so just working with these sequencing companies to let them know that based on everything that's been reported from the natural history and from the literature, that here are things that BBSOAS is known to be involved with. Additional resources for faster diagnosis. So some of these are tools that clinicians can use. That's those, the face match and face to gene. It's actually really cool tools where you take a picture of the child's face, and then based on some of the subtle dysmorphisms, so like different features, AI tools can learn and match that up with other people with BBSOAS and suggest to the clinician that that might be a diagnosis to consider. And then there are other sequencing and companies out there. Some of these are trying to identify patients way sooner. So some of these, such as Beginnings and Project Find Out, are in infants and babies. So instead of waiting potentially years and years and years of the genetic the diagnostic odyssey to get that diagnosis, some of these programs are trying to get the genetic diagnosis for infants with their very first symptoms. And then regarding what people who have been diagnosed and interact with the foundation can do. Matrix, as I've already harped on, and trying to fill out all those surveys. And then also just like publicizing BBSOAS, fundraising, just sharing anything on Facebook. You just help to raise awareness for this rare disease. And the more people that know about it, the more people will get diagnosed, and the more that researchers will be interested in the disorder. Also getting a CRID, a clinical research ID, which I think we'll be talking about tomorrow. Somebody, maybe? Yes. Okay, so that is just a way to make it easier for researchers to use the data that you put into Matrix, as well as associate that with data from the biorepository or any other research projects that you participate in. And then biomarker development is also incredibly important, so that's been discussed a bit as well. And so for this, it would be looking at proteins from the blood or urine to identify any special signatures about BBSOAS or anything that correlates with the severity of a patient. So, when a treatment is identified and is in clinical trials, brain development can be slow, and it's really expensive to have a long clinical trial. And so it might take 6 months to get a really noticeable change in improvement or with a communication, or with the CVI or something. It might take months before you can really definitively show that there's improvement there from the treatment. But a biomarker would be just from a simple blood draw, looking at proteins that researchers have determined correspond with, say, the severity of the CVI. And so this would make clinical trials much faster and cheaper, and so it would be easier to determine, what is going to be a successful treatment. And also EEGs could be another powerful biomarker. Combined Brain is working on setting up a EEG repository. And if any of you have EEGs that have been obtained from your children, if you can try to get that from your neurologist and just have that saved on a flash drive or your Google Drive or anything like that, then when Combined Brain has the EEG repository set up, hopefully later this year, then you'll be able to upload that. And then again, this serves as a central database for any researchers to be able to look at that, and they can look specifically at your child and what sort of different patterns to their brain waves that they might have. And then something that I've been working on is trying to identify which other groups to partner with, as the question from the person here had been about. So this is an in-depth process because it requires me to read basically everything in the literature about NR2F1 to identify what genes are being regulated by NR2F1, and then see if there's any disease associated with those other genes, and then try to link up whether there could be any collaborations there. I did find a paper so far that was looking at two related syndromes, this Waardenburg syndrome and Hirschsprung disease, that can be caused by potentially overexpression of NR2F1. And so that is basically the exact opposite of BBSOAS, when there's not enough NR2F1. And so potentially researchers studying these disorders might be interested in NR2F1 or with BBSOAS to see what is different or what's the same, and also partnering with the individuals for fundraising for these researchers. And then I'm trying to find more groups to partner with as well. And then there are also some of the larger umbrella groups that are not so much disease specific, but more looking at the phenotype as a whole. So there's Safari and the Simon Searchlight for autism, various epilepsy foundations. I think we might have submitted a grant with Cure Epilepsy. Yes, Jen is saying yes. So trying to leverage some of those larger groups that have way more funding to do research for BBSOAS, then vision impairment foundations as well. Basically lots of groups that could potentially help out with rare diseases like BBSOAS. And then looking into some more of the collaborations with scientists and researchers, collecting standard assessments for the natural history studies across all of the current studies and future ones, so that they're not splintered into different silos. You want to be able to directly compare patients from the US cohort with patients in Europe and any other studies that might be done down the road. So having kind of a standard assessment can be really helpful there. We've seen with other rare diseases that sometimes everyone has their own way of studying things, and so you have a really great group of data here on 20 patients, but then you can't compare it to the 15 over in this other study. And with a rare disease, it's really important that everyone is able to pool their data together. There's the Clinical Center of Excellence that Dr. Viral Shah has at Cincinnati, but then we might need additional ones down the road in other locations for clinical trials. Matrix, you all are utilizing currently, and that's another great tool. But one thing that we could try to push as the community is to have researchers use this existing matrix data. And then continuing here is to share all of the established models in a centralized repository. So especially for the in vitro models across institutions, it can be a very long, slow process to share cells from one lab to another. I've personally seen that draw out over the course of a year from two researchers, very willing to collaborate, but then just the legal departments of universities slowed things down a little bit. And so having a central repository like the Combined Brain Biorepository can really help to speed things up, because Combined Brain, we just want to share all these samples. Like, our legal department consists of people like me looking over it and being like, yep, I think that looks good, and sharing the samples from there. And so this is not just the patient samples, but this is also some of those research tools I mentioned earlier, such as iPSCs, and then it just makes it easier for other researchers who are interested in using those to obtain them. Additional things that the community could do is to host Zoom meetings for everyone researching BBSOAS to help them make sure that everyone is on the same page together and is learning about each other's work, as well as potentially bring researchers who are studying NR2F1 in other contexts together in a yearly virtual symposium to try to leverage any work being done outside of the neurodevelopmental field. And continuing with industry collaborations, so In Lorem is a nonprofit that creates ASOs for rare genetic diseases. We currently have an In Lorem application in progress for Melissa's child, Haley. And those ASOs will then potentially be useful to everyone else once the initial proof of concept is done. Multiple other strategies, there are companies and labs doing work there, so we could try to partner with some of them, as well as leverage the connections for the other manifestations of NR2F1 mutations. Then looking at outcome measures, so again, this is how do we know if a treatment is working. The outcome measures are one way of doing this. So the first step is to have a conceptual model. I mentioned this in the previous talk, where this is trying to identify every single symptom that has ever been seen in BBSOAS and understand what is it that is most important to the patients and to the families through interviews, instead of relying just on what clinicians reported in the literature. And so then once we have that list of all of the phenotypes and symptoms that are seen, the community can then kind of decide together what are the most important. And from there, you identify that, maybe it's the seizures that are one of your greatest concerns, or maybe it's, just the vision problems because it affects your child's independence and ability to just get around the house on their own. And then, so you identify those areas of greatest concern, what is it that would make the most impact on your and your child's quality of life? And then we identify the appropriate outcome measures to detect changes there. And this includes validating the observer-reported measures like the ORCA, then also EEG as biomarker, and also analyzing the existing data on matrix. So this could be, so that data is sitting there. Some of the uses of fundraising might be to supply a small seed grant. So this would be just a small amount of money to incentivize a researcher to take up a new project. And so once they have this preliminary data, then they can apply for larger grants, such as through the NIH, but they often need a little bit of help starting a project. So that's another role that you as the community can help out with. And then some additional experiments that could be done. So as I mentioned earlier, the endogenous ligand for NR2F1, so what it is that is activating NR2F1 in the cells is not yet known, and that's a really important piece of information that could affect what sort of treatments we use for NR2F1. So identifying that and other regulatory components, basically the things that are influencing the activity of NR2F1, and then perhaps those could be tweaked to adjust how the symptoms of BBSOAS manifest. And then especially looking at the other NR2F component proteins, NR2F2 and NR2F6. And then we want to test C26, which was that preliminary compound identified by Julio. and high throughput screens for repurposing compounds. And then additional experiments that should be done in animal models, such as the mice, is identifying a therapeutic window, if there's a certain age or period of development when BBSOAS can be best treated. So that's something that needs to be studied in mice. And then looking at overexpression of NR2F1, this is important for gene therapies because gene therapies can sometimes result in excess of the gene as you're trying to modulate how much is there, and so determining whether this is harmful or not. We also need to understand the role of NR2F1 in the adult brain and determine which neurons and what regions of the brain are expressing it. And then things kind of as the foundation as a whole can help guide the entire NR2F1 field across researchers and various projects is to establish the research priorities and provide funding for that, and establish a formal request for proposal program. So this would be inviting researchers to submit grants to the foundation, and then the board members can meet and decide what it is that is most helpful for the community and provide funding there. And this would be likely small seed grants that help a researcher to just kind of kickstart a project so that then hopefully they'll be invested in it for years to come and can get the much larger grants from funding bodies like the NIH that they just couldn't have gotten otherwise without having that initial seed money to have the preliminary data. Because you can't just write a grant to the NIH saying like, I have an idea. You need a little bit of proof of like how, showing that you are able to investigate this and that your idea seems to have like good preliminary work, and that's expensive, so the seed grants from the foundation can help a lot with that. And so that's kind of just overview of everything that I've talked about here, like all of the things that we could be working on. For example, like the faster diagnosis is something that's important to get more patients involved. More patients means more people to participate in all of the projects like Biorepository or Matrix, and to help with fundraisers for all of these other projects. Then things that we do have, like for example, there are multiple mouse models, and so that's a great thing that the foundation has. The research symposiums such as this and the one that was in Denver in the fall are amazing opportunities. Then things that we still need to do include the EEG analysis and a repository for that. So yeah, that's kind of the overview that I've been putting together and trying to identify all of these researchers in the field. And yeah, so I'll take any questions on that.
Thank you so much, Sarah. Questions. Also, just to note, if you're participating online, if you can write your questions into the Zoom chat box. We know that a few questions went into the Hoover app. We've got those from the last presentation, and we'll share those with Sarah. But if you've got questions online now, please put them into... Oh, we've got some questions, actually. Why don't we go to the online ones before we come to the room, just to keep it fair. Okay, so there's a question on muscle tone to understand what research has gone into low muscle tone and understanding the impact.
Yeah, so I don't think there's been any research specifically on how NR2F1 mutations result in low muscle tone. It is a common symptom that is seen across many genetic neurodevelopmental disorders. So it seems to be that there are some common underlying pathways where just due to the altered activity of the neurons in the brain and potentially in the spinal cord, that just the signals that are being sent out to the muscles are different, and there's not enough. But regarding NR2F1 specifically, how it's resulting in that, I don't think there has been any research.
Thank you. We've got a question on EEG. I'm not a scientist, but I've got a thought on this. Is the EEG only helpful if there's an epilepsy diagnosis?
No, it can be helpful for anybody. So I'll give an example. Angelman syndrome is a genetic neurodevelopmental disorder that does not have seizures in the majority of patients. Most patients have no seizures at all, and then some have like just a handful in their entire life. But there was a biomarker identified from their EEGs. Like there's a specific pattern, like change in the pattern of their brain waves that had nothing to do with seizures. But they found that patients who were the most severely impacted had more of this pattern, while patients who were less severely impacted in all other domains, the motor coordination issues like verbal communication, et cetera, all of that was associated with a less severe alteration in this specific pattern. And I've also heard projects for other developmental disorders where they're seeing perhaps some similar things. Nothing's been validated yet for these other ones, but it seems to be a strong component for any of these neurodevelopmental disorders is that when you are able to look at enough EEGs, specific patterns stand out and that have nothing to do with the epilepsy. It's really just how the kid's brain as a whole is being altered by the disease.
Thanks, Sarah. And last one as well from online. Can patients provide a document that would force sharing of samples across research institutions for the purpose of better collaboration and accelerate research? I think that's going to come in patient registry with CRID. But would you have anything? Is that
Yeah. So when you sign up for the Combined Brain Biorepository, there is a consent form that asks you whether you are comfortable sharing the data and the samples across different institutions, and so you can say yes or no. And then also the CRID is an anonymous identifier linked with your samples and your DNA that makes it easier for researchers to associate the samples with all of the data you provide.
Thanks, Sarah. We've got a great presentation. We had CRID recently do a webinar, so that's available as well. I'm going to turn to a couple of questions in the room, and then I'm conscious of time that we've got Dr. Shah waiting on Zoom for us as well. So Meredith.
So it took us many years to get the occipital lobe epilepsy diagnosis. Is it the most recent EEG, or is it all of the EEGs? Because we went to hospitals, we went all over the place, and none of them would identify that she was having seizures, even though we saw the seizures ourselves. And so it's almost like, with that past history, even though it wasn't identified then, to see what those brain patterns were then, because once we finally got the diagnosis, other parents started reaching out saying, well, my kid was doing that too, maybe we should get an EEG. And it's that occipital lobe epilepsy part that I think is kind of that it happens so deep in their brain that they're not showing up on EEGs.
Yeah, so all of those EEGs would be helpful in part to have the evidence of things like this. Of clinicians and neurologists need to look deeper. They need to look for these occipital lobe seizures. But then also there might still be patterns in the rest of the EEG that weren't popping up initially just as like a one individual, but then if there's a neurologist looking at specifically 50 different EEGs from BBSOAS patients, they might notice subtle differences that then they can try to validate, and so they might be able to find still useful things from even just some of those regular EEGs that didn't require nearly so much effort to get. Yeah.
Hi, I'm just curious because it's been mentioned about the facial dysmorphism in a couple of the patients. What are you referencing? Because most of the kids I've seen look typical to me. So
Yeah. So most do look typical, but there can be some subtle changes that, again, it's only through looking at many different patients and having the AI tools, because it might be something really subtle, just like a change in the eyebrow shape, where it's like it was still within the realm of normal, but if ever many of them have a specific shape that it's like, well, maybe that's part of like just the development with BBSOAS. So yeah, it's not like strong facial dysmorphisms, but just one of the many opportunities that by having these large data sets, we can discover things that previously we had no idea were patterns, similar with the EEGs, where it might look normal until you see 50 of them all together.
That would be like the Cincinnati thing that you said that would be it?
No, I don't think that anyone at Cincinnati at the Center of Excellence is looking at specifically that. This one is more of a tool for other clinicians who might be trying to diagnose various neurodevelopmental disorders to use these tools, and potentially there might not be a unique signature for BBSOAS, but we don't know until we try. And so it has been really helpful that there are certain syndromes where even parents have been able to diagnose their child just because they've stumbled across pictures of other kids with the syndrome online, and they're like, oh my gosh, that looks exactly like my kid. And then they go to their neurologist, and the neurologist is like, oh wow, you're right. And so it's one of those things where it's like we won't know until we get the data to show. Yeah.
I just wanted to speak to that, Yolanda, right? The facial dysmorphia. I know when I read that initially, a while back, I thought, my daughter doesn't, that's pretty harsh, But again, it's a technical term, and I wish they could change it, but it is what it is. And I think of it as, especially with our population, I see it personally as my daughter, I feel like, resembles physically a lot of the other patients more than she resembles us, her parents, or her family members. She looks pretty different than her sister, for example. She looks more like Meredith's daughter, Lola, than she does. So that's just an example of what they mean by facial dysmorphia. I think and a lot of families, parents, we've talked about this, that there's just something, they just have this something that they all sort of have this sort of look. Mm-hmm. It's a beautiful look, but there is a pattern, so.
Okay, we've got one more question in the room, and then we're going to have to...
That the term dysmorphia, it sounds terrible, but it really only means different form, right? So if you look at it from that aspect, it just has like a negative connotation, but it just means something is shaped differently, And that way you don't take such offense to it.
Right, yeah, like there are some disorders where it's like the patients tend to have thin lips, not in any sort of like abnormal way, but it's like regardless of what their parents may have had, like everyone has like very different lip shapes, and then these kids, like, they look normal, but some of them, as Carly was saying, it's like, actually they have way thinner lips than their parents, and so it's not like that they're like completely disformed, it's just differently formed.
Sarah, thank you so much. That's really fantastic.